All right, hello everyone. This is for February 22nd and 23rd. So we're going to start with our Black Scientist of the Day, and I've rather enjoyed these, so I might keep them going past Black History Month and just do Scientists of the Day, because why not? What I want to do real quick after that is do a review over genetics and Punnett squares, so just making sure that you remember all of the vocab and concepts that we've covered so far. Then we're going to jump into when things are a little more complex than just what we've been working with. So we're going to talk about non-Mendelian genetics, specifically codominance and incomplete dominance today. I have a practice assignment that we are going to do, and then we're going to end with an exit ticket. Starting off with our Black Scientist of the Day, we have Dr. Patricia Bath, born in 1942, and she unfortunately passed away just a couple years ago. So Dr. Patricia Bath was an American ophthalmologist and laser scientist. So an ophthalmologist just focuses on the eyes. And she became the first female ophthalmologist to be appointed to the faculty of the UCLA School of Medicine, Jules Stein Eye Institute, in 1947. Then she became the first woman to chair an ophthalmology residency program in the United States. So residency is when medical students have to work or get experience as residents. So she was the first woman to chair that ophthalmology residency program. That was in 1983. And then she became the first female African-American physician to receive a patent for a medical invention. And she did that in 1986. So before she did all of those firsts, or a lot of those firsts, while she was completing her medical training in New York City in 1969, she noticed that there were far more blind or visually impaired patients at the eye clinic in Harlem as compared to the eye clinic at Columbia University. And she was curious as to why. So she conducted a study and found that the prevalence of blindness in Harlem, why there was so many issues, was because they lacked the access to eye care. They just did not have that kind of medical attention. So to solve the problem, she proposed a new discipline called community ophthalmology, which trains volunteers to offer eye care to those populations that just don't get the access to eye care that they need. And that concept is now worldwide employed and has saved the sight of thousands who would have otherwise gone undiagnosed and untreated. And then lastly, her research on cataracts, which is a certain disorder of the eye, has led to a new invention of a new method and device to remove those cataracts. And that was called the laser phaco probe. And she, she earned a patent for that technology in 1986, and it's still used today. So that's pretty incredible. Our Black Scientist of the Day today, Dr. Patricia Bath. All right, so the next thing I want to do is review some of these genetics concepts that we've been talking about. So I'm just going to pull up this basic slide and talk to you or remind you a little bit about all the different things we've talked about throughout the last 6 weeks, 7 weeks. So as a reminder, each of those letters, the big T's and the little t's, they are called alleles. And those are different versions of a trait. So if we're looking at height, you can be tall or short. Those are different variations of that trait. And we have dominant and recessive alleles. Okay, dominant just means that we see it more often and it tends to mask the recessive. So don't forget about those. And then when we're looking at these words, genotype and phenotype, genotype refers to the genes, the gene combinations, the allele combinations, allele combo. Okay, and what we've talked about in Mendel's genetics is that we can either be homozygous dominant, heterozygous, or homozygous recessive, okay, depending on if you have 2 dominant alleles, 1 of each, or 2 recessive alleles. So that's genotype. When we look at phenotype are physical characteristics. And usually it'll tell us in the word problem what physical characteristics we're looking at. Physical characteristics. Okay, for example, here we have tall and short. Those are the different phenotypes that we can have. Now, what this is right here is a Punnett square. So as a reminder, Punnett squares help us calculate the probability of a certain trait getting passed down from parent to offspring. So for example, here we have two parents, one that's homozygous dominant for tall and one that's homozygous recessive for short. So essentially here we're asking the question, if these two individuals made babies, what's the possibility of them being tall and what's the possibility of them being short? Okay. So as a reminder, how to do these Punnett squares, we'll put one parent at the top and one parent over on the left. And you're just going to bring down the alleles from the top and bring over the alleles from the side. So then you will have a big T from the top, little t from the side, big T from the top, little t from the side, big T from the top, little t from the side, big T from the top, little t from the side. Okay, so these are your genotypes on your Punnett square. When I ask you to give me a genotype or phenotype percentage, the first step should always be to list out all the ones that you see in the Punnett square. So we're talking about everything that's in this box right here. So remember, genotypes look like these. They're combinations of letters. So we have these 4 genotypes. Now, these 4 genotypes are going to be the exact same, right? They're the same thing. They're all heterozygous. So for genotype, I'm just going to put that one because that's the only one we got. If we got 2 or 3 different ones, I would write them all down. But since they're all heterozygous, that works for me. And remember that an individual box is worth 25%, right? 25, 25, 25, 25, make 100%. So since we have 4 boxes that have the same genotype, 1, 2, 3, 4, 4 boxes out of 4 is going to give us 100%. Okay. And what we have been talking about up until this point is when we're talking about phenotypes, it's going to be either or. So our only options here are they have to be tall or they have to be short. That's it. Okay, so since they all have the same genotype, they're all going to look the same as well. So it's up to us to figure out if it's going to be tall or short. So in this case, because it's one option or the other and no in between, we're going to go with what the dominant trait is. The dominant trait tends to mask that recessive. So it's almost like this one doesn't exist because all we see is this. So because big T is tall, we are just going to see tall plants. And just like the genotype, because we have 1, 2, 3, and 4 boxes of 4 that are all going to be tall, that means 100% of our babies are going to be tall. So that was some just quick review on our vocab and how to do our Punnett squares. So hopefully you have this because we're going to work with the same kind of idea, just introduce different things. Okay. So now we do a little bit of notes, nothing crazy, but I'm going to introduce to you this idea that Mendel just didn't know everything, which makes sense because he was around the 1800s and technology has changed a lot. So before, when we're talking about traits, we've talked about how they're either or. You're either black or brown fur. You either have brown or blue eyes. You're either tall or short. And even while we're doing these examples, a lot of you have had questions on, well, there's other hair, there's other colors, there's other eye colors, there's other heights than tall and short, there's in-betweens. And you're right. So what we've learned is that Mendel's laws of inheritance don't apply to every single trait. So that's what we're going to talk about in the next 2 weeks. So Mendelian genetics is essentially complete dominance. And what we've learned so far, what Mendel has taught us, is that there's only two possible phenotypes. There's either the dominant one or the recessive one. So in this example that we just did, we have a tall plant and a short plant that come together to make babies, and they don't make a medium-sized plant. They either have to make tall or short. So they'll make tall plants. And this is Mendelian genetics. This is what we've been working with. What we've realized is not all inheritance is based off of those rules. There are other rules. And that's what's non-Mendelian genetics. That's what we call non-Mendelian genetics. So when we have different colored flowers or cats with different colors of fur on a single individual, or variety of eye color, and even blood type are all traits that don't follow this dominant or recessive rule. So again, Mendelian genetics describes it based off of complete dominance or recessiveness, but we have other types that Mendel hasn't even considered. So these are 5. Hopefully I will hit at least 4 of them in the next 2 weeks. We have incomplete inheritance, codominance, multiple alleles, sex-linked, and polygenic traits. We're going to talk about the first 2 today. So starting with incomplete dominance. In incomplete dominance, the hybrid or the heterozygote shows a third or blended phenotype. Okay, so this is that in between. I'm going to write that on here actually, just to remind us all. It's the in between. Okay, it makes up a whole new phenotype. So for example, if we have these 3 flowers, we have a red one and a white one. So let's look at red and white. Okay, if a red flower came together with a white flower and came together to make a pink flower, that would be an incomplete dominance inheritance pattern, because pink is an in-between. You don't see the red, you don't see the white, you see this mixture, this blend of the two. Kind of like if you took red paint and added in white paint, you'd get pink. Another way I like to think about it is it's incomplete dominance. So it's kind of like it started off white and it wanted to become a red flower really, really badly. And it was trying so hard, but it couldn't make it. It got stuck. So it's not quite white and it's not quite red. It's in the middle. So that is an example of incomplete dominance. Height is another example that would kind of follow this. You're not tall or short, you're kind of anywhere in between. So you can get like medium-sized or medium heights. So what I want to do is an example of a Punnett square with this idea, with this inheritance pattern. So here we are. So we have a red flower that we're going to cross with a white flower. So let's say that we're going to use R for red and W for white. So we're going to take a homozygous red and a homozygous white, and we're going to cross it and get the genotype and phenotype percentages. So just like usual, we're going to put it in a Punnett square. So we have our red up here and our white over here. So you bring them down, bring them over. So you're going to have a big, or you're going to have an R from the top and a W from the side, R from the top, W from the side, R from the top, W from the side, R from the top, W from the side. Okay, so you might notice that all 4 of those genotypes are the exact same. So for genotypes, we just have 1 to include, right? So we have our W, and they're all the same. 1, 2, 3, 4 RWs, 4 boxes out of 4 gives us 100%. Okay, so up to this point, pretty much everything is the exact same. When it gets to phenotypes, it's where it's weird. It's where it gets a little bit difficult. Okay, so previously we were taught that phenotypes are 2. We either have it or we don't. We have this one or that one. In incomplete dominance, we have a third one. So we have a red flower and a white flower that came together to give us a blend. So if we take red and white, if we take those colors and mix them, what do we get? We get pink. Okay, it's a blend of those two colors. And just like the genotype, because there's 1, 2, 3, 4 boxes of 4 that are that phenotype, it's going to be 100% pink. Okay, everything very similar. The only thing is, how do they come out physically looking? How are they different? All right, other than incomplete dominance, we also have codominance. In codominance, both of the alleles for the trait are dominant. There's not one that is dominant over the other. They're both just as dominant. So instead of the heterozygous showing one trait, it'll actually show both. For example, here we have a white bull and a brown cow. Okay, so when they come together individually, this one is fully white and this one is fully brown. But when they make a baby, you see the white very clearly and you see the brown spots very clearly. So it's not like incomplete dominance because we don't see a blend of them, right? We don't see a whole light brown cow. We see a cow that has both traits from the parents, sees both the white from the bull and the brown from the cow. Okay, we can see both very, very clearly. And that is codominance. So again, incomplete is when it's a blended version of both. So it makes an entirely new phenotype. And codominance is when you can clearly see both traits together. Now I'm going to do an example of a Punnett square with codominance. Very similar thing. So here we have a white rooster that's crossed with a black hen, and it gives us the parental, and it gives us the parental genotypes here. Boom. So we have our WW for white and our BB for black. And again, we'll just bring down the alleles from the top, bring over the alleles from the left, and we'll get W from the top, B from the side, W from the top, B from the side, W from the top, B from the side, W from the top, B from the side. So again, 4 exact same genotypes. So under genotypes, this is what we got. We got WBs, and that's all we got. So again, 1, 2, 3, 4 boxes out of 4 is going to be 100%. Now, remember, codominance is when you see both of those traits very clearly. So the phenotype that we see, because we only see one, okay, they all have the same genotype, so they all have to have the same phenotype. It's not going to be either black or white. It's going to be a combination. I have a picture on the next slide. Well, I kind of just showed it to you, but regardless. So you kind of have a black and white chicken. Okay, black and white. So you'll see black feathers and you'll see white feathers. You'll see both very distinctly. And of course, because all 4 of them have the same genotype and therefore will have the same phenotype, 100% of them will be this black and white color. I'm going to show you that next slide. Bam. Okay, so it has some white feathers, it has some black feathers. You can clearly see both traits very visibly. It's not gray, it's not that incomplete or halfway phenotype. It is distinctly both that you see at the same time. So those are the 2 that I wanted to focus on today, incomplete dominance and codominance. And with this information, now we can start on the practice assignment. So from here, we're going to go to Canvas. So from here, we're going to go to Canvas, and you're going to go to our modules as usual. You're going to scroll all the way down. We are in week 8, so all the assignments are going to be right underneath there. And you can take a look at the incomplete and co-dominance notes if you would like, just because I do include the definition and some examples in there. But the Punnett square examples we did together are going to be blank, but fortunately you have this video to show you how we work through them. The incomplete and co-dominance practice is the assignment that we're doing. So you're going to use CAMI for this as well. So here are the instructions for CAMI. As a reminder, you'll download this document to your computer. So keep clicking until something pops up. So something pops up at the bottom, but for your Chromebooks, it'll pop up on the lower right-hand side. And then you'll go to CAMI. Sign in. Open from your computer, and it should be one of the most recently downloaded things. So you'll just, bam. All right, so this is what it will look like, and it's just one page. The top has a table, and it's just to be there to help you to differentiate between incomplete and co-dominance. So using the notes we just did, you can fill in the boxes. So under incomplete dominance, oops, you will give me the definition. So what is it? And then what does it look like? So give me an example. And then for co-dominance, you're going to do the same thing. Give me a definition and an example. And for these two questions down here, you have one that is going to be incomplete dominance and one that is going to be co-dominance. So you just have to figure out which one is which based off of the definitions you put here. So they're going to be pretty simple. They give you a problem. For example, the first one is you cross a pair of all black Jordans with a red pair of Jordans. It gives you the genotype. And they create a black and red spotted pair of Jordans. So you're going to tell me what type of non-Mendelian trait is it? Is it incomplete or is it co-dominance? And then you're going to create a Punnett square to tell us what kind of genotypes could you get when you cross a black and a red pair of Jordans. So off to the side, you'll do your Punnett square. You'll tell me each genotype you get and the percentage of each, and each phenotype you get and the percentage of each. You do that for both of those. If you have any questions about these, don't hesitate to let me know, and I will help you out as best as I can. After you're done with this, you are going to go back to Canvas, and the last thing we have is that exit ticket. That exit ticket is going to be right underneath this practice assignment. It's called Non-Mendelian Exit Ticket with today's date. And just like our other exit tickets, it's a quick 5 questions. So we have the statement. It says pups of long-tailed and short-tailed parents often split the difference and have medium-length tails. So the first question you're going to answer is it incomplete dominance or co-dominance? And how did you know? So again, going back to those definitions we just talked about. For number 2, it asks if the parents' genotypes are these, LL and SS. What would the genotype be for a medium-length tailed dog? Number 3 asks us to create a Punnett square for these 2 individuals. Please make sure that you are using these 2 individuals. Don't just go off of number 2. And then show me. So you can either do it on your own and take a picture, write it in the text box, however you want to show me that Punnett square. And then 4 and 5, give me the different genotypes. So you don't necessarily have to include percentages, but if you do, I will give you extra credit point because it's always good practice. And then for number 5, what are the different phenotypes that you get? Again, percentages are extra credit. And again, please make sure that you are reading these questions carefully. Read them thoroughly. Don't skim and assume that you know what they're talking about. And keep in mind that 4 and 5 are asking for distinctly different things. So if you have any questions on what genotypes and phenotypes are, there's plenty of stuff on Canvas to remind you and Google. So don't be afraid to look it up. And once you're done with this exit ticket, you are done with what we did today. And again, if you had any questions at all, please don't hesitate to let me know.